This week on Careers in Discovery, I'm delighted to be joined by Mike Stubbington of CS Genetics. Mike, welcome to the show.
Hi, Tom, and thanks a lot for inviting me. This is the first podcast I've ever done, so that's pretty
Oh,
exciting.
really?
Yeah.
Well, welcome. We'll be gentle. um, we always start, Mike, by talking a little bit about what you're up to now. So I'm really interested to learn more about CS Genetics and single cell genomics made simple. Tell us a bit about what you're up to.
Sure. So I'm Chief Scientific Officer here at CS Genetics, and that means that I lead essentially all of our R&D efforts as we
Hmm.
come towards building our first product. And yeah, as you said, we're building something that's really trying to make single cell genetics approaches very simple and democratized, and to really open up access to areas of science and people doing science to use single cell that haven't existed up until now. Essentially what we're building is it's probably been inescapable over the last decade or so that single cell approaches have become incredibly powerful
Yes.
for revealing biology, understanding of cell types and cell states, and all sorts of things in biology, and they've been amazing. But they've always required, instrumentation or complex workflows, and they've been expensive, and
Mm,
I think that's restricted who can use them and in the context they can be used. So we're building something that's entirely instrument free. There's no, you don't have any capital expenditure up front to buy any kind of
okay.
box or instrument. So it's just reagents that we will sell, and everything's done in very standard laboratory plasticware. So it's a very simple approach. We're actually calling the product Simple Cell, which isn't a
Okay.
name I came up with, but I think it encapsulates what we're doing really quite nicely.
Yes.
And it basically is a very quick and easy workflow. There's no complex pipetting steps where you split samples and then bring them back together. It's all very kind of easy for someone to do, and it also the entire process scales using exactly the same assay from one sample done by, say, a grad student on a bench up to eventually, we believe, thousands of samples done hopefully by automated liquid handlers in a much more kind of industrial setting. And
Mm.
we're very excited about all areas of that spectrum, just in terms of enabling experiments that aren't being done today.
Yeah, I see. And a lot of people listening to this will be very familiar with single cell genomics, single cell sequencing, those sorts of things. But for those who aren't, could you give us a brief intro to why it's so powerful and also why it's been so complex?
Absolutely. So I guess until this era of single cell genomics, if you wanted to understand something about a cell, and let's use gene expression as an example, because that's the
Mm.
sort of the first one that everyone has looked at, what genes are switched on in a cell type of interest. What you would do is you would somehow purify that cell type of interest as best as possible. Let's say you're looking at T cells, you might do flow sorting or some other approach to purify a population that you think is representative of your T cells, and
Hmm.
you'd have tens of thousands, hundreds of thousands of those cells, and you would, in bulk, ask your question, what genes are switched on in this entire population of cells? And that works. I mean, we learned a lot of biology before single cell, so obviously
Yeah.
that works to an extent, but it doesn't get at any heterogeneity that's present. Every cell is its own unique thing. Even in a population that you think is pretty homogeneous, it's not going to be. There are going to be differences. And so what single cell does is it says, well, actually, what I'm going to do is I'm going to ask that question, what gene is expressed in every cell individually in my population, and then I'm going to use computational methods to understand the heterogeneity that's present. And the
Okay.
kind of two analogies that we always use. One is, if you were asking the question, what color are my cells, and half of them were blue and half of them were yellow, in a bulk approach, you'd say everything was green because it
Right,
averages the
right, right.
signal across everything.
Yes.
The other analogy that's very, very popular is like trying to understand what's in a fruit bowl by making a smoothie and tasting it if you do
Okay.
bulk approaches, whereas if you do single cell, you just pick the individual pieces of fruit out of the fruit bowl and look at them individually.
I see. So greater accuracy, but also greater nuance and depth.
Absolutely, and it tells you, you can also take a heterogeneous population of cells, just white blood
Right.
cells, and look at the entirety of the system there, which is really cool. You can identify cell types that you didn't know were present. You don't need that a priori understanding of what's there to purify the cell type. You can go in and look and see what's there.
Yeah. Okay. Interesting. And then, so you mentioned that obviously to take these approaches currently, there's a lot of expensive equipment involved and it's complex methodology and all that kind of thing. Uh, Without giving away the family secrets, tell us a bit about that simplification that you've done with it.
Yeah, absolutely. So we have an approach which really doesn't rely upon this idea of partitioning. So if you look at single cell technologies up until now, they always put a kind of physical partition around cells at some point, whether that's in a droplet where you have the outside of the droplet around a cell, or in some of the methods where you split cells up into different wells, then do something to label them, then pull them back together, and then split them again. Those physical walls of the well are essentially doing the same job where they're physically
Mm,
separating cells. We have a method that uses an entirely different approach where we have a reagent that binds to cells and does the kind of indexing of the oligonucleotides that are present in the cells. And we have another reagent that really drives that single cell behavior even in solution phase, and we call this kinetic confinement, and we've talked about this and in other places. And really what it does is it uses viscosity of a buffer to really favor cell indexing interactions that occur
mm.
in those cell reagent pairs and not in the wider population, and that gives you that single cell behavior.
Yeah, I see. So complete change of fundamental experimental design to get to this point. Okay,
Absolutely.
interesting. And then, so tell me, Mike, a little bit about in this sort of company at the stage that CS Genetics is at as the CSO, where is your focus? Where are you spending your time? What does your I'd say what does your day look like, but probably more accurately, what does a week look like typically for you?
Yes, so a lot of my time is really helping the team figure out prioritization. You said, at the stage of our company, we are not a company with huge resources at the
Hmm.
moment, and we're building this product. So we have to be very smart about how we spend our time, energy, money, sequencing space on our sequencer. And so really helping people work out what are the best experiments to do,
Mm,
what are the experiments that are going to take us on the shortest path to product launch, working with the team on one-on-ones, making sure everyone's happy, doing that sort of development work with the team, thinking about the experiments, looking at data. I love looking at data. That's always the best bit. If someone has something exciting, going to their computer and looking at that, that's pretty cool. Obviously, things working with the rest of the leadership team to think about, to make sure the strategy is right across the entirety of the company, across operations. I'm responsible for R&D, but we interface really closely with operations and how we're going to
mm.
manufacture the product to make sure we're staying really tight on that, looking at timelines. Sometimes I pick up a toolkit and go and do a little bit of maintenance in our building. We've just moved to a new building,
Fair
and
enough.
sometimes,
Yeah.
minor things need tightening up. And so yeah, it's pretty varied.
Yeah, absolutely. But you still get plenty of chance to sit with the data and be close to what's going on in the lab.
Absolutely. I love managing to open up a code editor and notebook and do some analysis myself when I get a chance just to really dive into it.
Fair enough. Well, with that in mind, we always like to sort of explore the origins of people's careers as well. And so really interested for you, Mike, why science, first of all, why biology, why computational biology, drug development, why this career for you?
That's a really good question. I think I've always been interested in science ever since I was a pretty young kid.
Hmm.
And I think it sort of became that sort of virtuous circle where I was interested in science and also I was quite good at it. It just sort of clicked with me, the
Yeah.
idea of understanding the world. And so then I got better at it and paid more attention to it in school. And it became a path that looked like it would be good for me. My parents were always incredibly supportive in the pre-internet days of making sure I had access to books and
Yes.
libraries and magazines about science so that I could, feed that interest. So I was always interested in it. It was always a career I wanted to do, and went and did my undergraduate in well, natural science at Cambridge. Cambridge has only one science degree, which
Right.
it's called natural science, and you choose what you want to do kind of within that. And I specialized in molecular biology and biochemistry towards the end. So that was
Mm.
always something that felt like I was on a path. I had some fantastic teachers at school. My GCSE and A-level science teachers at my just local state school went absolutely above and beyond to
Right.
help all of the kids in my year and associated years who were interested in science, and they would find us positions in the summer to go and do projects at Birmingham University.
Oh,
I've
okay.
spent, I think, 4 weeks doing a project at Birmingham University one summer in between my A-level years, which was
Mm,
entirely because my biology teacher was just enthused about science and enabling people to discover things. So
yes.
I just got to experience some of this stuff, and that showed me what a career in science could look like. Going into a lab, working with PhD students and postdocs and the PI, and
Mm,
doing experiments and being trusted to, I mean, looking back, they were pretty simple experiments. I don't know that I did them that well, but being trusted to do them and then having to write up the results and communicate them in a
right.
lab meeting. So I had those opportunities, went to university, continued to pursue science. And at this point, I wasn't very computational. I learned to code when I was about 12.
Mm,
My my parents bought me a computer that didn't really play games, because they knew I would just play games, and I mean they were correct. But it did have a programming language built in, and so
right.
in an attempt to extract maximum value out of this present that they gave me at Christmas one year, I read through the very thick programming reference guide that it came
Hmm.
with, and learned this programming language and started writing little programs and playing around with it. So I guess I got the sense of what it meant to write a program and some of those very
Yeah,
basic
I see.
concepts, but I wasn't really applying it at university. I was doing wet lab science and thinking
Mm,
about wet lab work. And then I did my master's at university, which was a 2-term long project in fourth year, where again I did actual research in a research lab
right.
amongst grad students, PhD students, postdocs. But I didn't stay and do a PhD. At that point, I was
Hmm.
kind of done with university. I wanted to go and do something else. I didn't have that burning desire to go and do a PhD, and probably that was the right decision, because I
Okay.
probably wouldn't have committed properly to the effort it requires. So I left and I joined what was then called the Health Protection Agency. So this is now
Yes.
called Public Health England. It got rebranded. I think it's been rebranded once before that as well
Hmm.
by the government, but it's a government body, kind of arm's length from the NHS, that I was actually working at Porton Down, which you might have heard of as the home of the UK bioweapons project back in
Yep.
the early part of the 20th century. Obviously, now it's not involved in offensive bioweapons, but there's a lot of work there. There's a military site and a health service site involved in protecting UK troops and the UK population from potential bioterrorism or biological warfare.
Mm,
And I worked for the health service site there, and we were looking at high containment infectious diseases. So I'd worked quite a lot on anthrax, for example, looking at
okay.
genomics in anthrax, sequencing different strains, doing some work on pathogenesis. And during that time, there was a desire to look at a bioinformatics problem, essentially. And I managed to get involved. We had a bioinformatician on site there, and I worked with him to help solve this problem. And I kind of regenerated in my brain all of this bioinformatics, or all of this programming that I'd learned like a
Right.
decade before, and thought, oh yeah, this is actually pretty fun. And so that reminded me, and so sort of started to get me into the world of bioinformatics and
Mm,
analyzing
mm,
sequence data and doing computational work. And then I went on and did a PhD. I'd actually started a part-time PhD while working at the Health Protection Agency, registered at Cambridge, actually with my old master's supervisor, but doing a different thing, involved actually in anthrax pathogenesis while I was working for the HPA.
okay.
And it became apparent to me that that wasn't actually going to pan out. I didn't the time, doing a part-time PhD, I think is really, really hard when you have a job,
Hmm.
and trying to fit it in and
Mm,
work on the resources, and you're sort of at arm's length from your university. So I realized that probably wasn't what I wanted to do, and actually a full-time PhD was going to be better. So I
right.
I went to findaphd.com, looked at what was available at the time, and applied and interviewed at the Babraham Institute,
Mm,
just outside Cambridge. And a fantastic group leader called Anne Corcoran, who was just an amazing mentor during that PhD time. Worked with her again in the wet lab on B cell development there, and we were using an assay that generated a lot of next generation sequencing data. This was in the still relatively early days of Illumina having sequencing machines after the Solexa acquisition, and generated a lot of sequencing data. And Babraham had a fantastic, still has a fantastic bioinformatics core of
Mm,
really talented bioinformaticians, but they're also super busy. They have a lot of projects to look, or when I
right.
was there, had a lot of projects to look at. And I kind of wanted to own my own analysis, do it myself, do it quickly, and also I was just interested in doing more bioinformatics. I did more and more informatic analysis, and by the time I was writing my thesis, I was having to be almost physically restrained from doing just another bit of analysis on the data because I had to actually write the thing and get it
Yes.
submitted. And it also taught me that I think I'm a better computationalist than I am an experimentalist. I never had those magic hands that some people had.
Mm,
There were postdocs I was working with at the time who everything they touched, the experiment worked beautifully. It was just super high quality. I never had that. I was okay, but it wasn't my like core skill. And I think
yes.
also when you're doing molecular biology, you're moving tiny amounts of colorless liquid around tubes, and then you're getting a readout. And if it doesn't work, you have to figure out why it didn't work, which of your colorless liquids didn't work, or what condition didn't work. And the sort of,
Mm,
if you like, the debugging loop of that, the iterative loop is super long and quite hard to figure out.
right.
Whereas what I really liked about computational work is you write a program, you run it, if it fails, you get an error message, which is more or less informative, and then you can
Yes.
try and figure out, did I miss the semicolon at the end of this line, or did I not close a bracket, or have I just taken an entirely wrong approach? And it just
Mm.
felt much more knowable, and the iterative loop was much faster, and that resonated really well with my brain. So I decided to then kind of pursue computational work after that.
That makes sense. And then you had the opportunity to go and work in one of the leading institutes that there is for someone who wants to learn all there is to know about bioinformatics.
Absolutely. So I joined Sarah Teichmann's group at the European Bioinformatics Institute. And
Hmm.
she actually had a joint appointment at EBI and at the Sanger Institute at the time. And they're a couple hundred meters apart at the
Yeah.
Hingston Genome Campus just outside Cambridge. Yeah, and when I was talking to Sarah about joining her group, we initially talked about doing a kind of joint wet and dry lab postdoc, and I do a bit of both.
Mm.
While I was writing up, Sarah gave me a call and said, actually, she didn't really have enough lab space at the time, so would I mind just starting as a dry lab postdoc? And I actually thought, yes, that sounds fantastic
Yeah.
because I'd really like to focus on that. So I joined Sarah's group at EBI, and EBI is a fantastic, like you said, it's an amazing environment to do bioinformatics, incredibly well resourced in terms of compute, just an amazing set of people to talk to and learn from, which I found just very and potent for my development. And Sarah was working at the very early stages of single cell biology. She had
Mm.
an instrument called the Fluidigm C1, which was, I think, the first sort of instrument you could buy to do single cell approaches. People
Hmm.
before that had done, the first single cell papers were mouth pipetting 9 cells into individual wells of a plate, which is
Right.
kind of amazing. And then the C1 would do 96 cells at a time, or somewhere between 50 and 96 cells at a time. So Sarah had one of these. She was looking at T cells as a model
Hmm.
of cellular differentiation in single cell. And again, like using that ability to look at every individual cell to understand what's happening in the differentiation process and the commitment process to in a T cell response is amazing. And I realized that you could extract more information from those data sets to look at the T cell receptor sequence. And I won't go into the like the deep biology of this, but basically
Sure.
every T cell, when you make T cells in your thymus or when you make B cells in your bone marrow, the DNA that encodes the bit of that cell that will detect antigens and detect foreign stuff as part of the immune response, that's this amazingly complex kind of cutting up and shuffling around of DNA segments to give you incredible diversity of
Mm. Mm.
sequence. So your immune system can recognize anything it might need to recognize. And I realized that you could extract those very variable sequences from the single cell data by playing around with some computational approaches. And I'd looked at that in my PhD on the wet lab side, and then coming back to it from the dry lab side with Sarah, wrote some code to look at that. And so it was kind of extending the single cell analysis beyond just gene expression to something a bit different.
Right, I see. So really making the most of this brand new technology at the very sort of birth of it. Um,
Absolutely.
which I imagine there was parts of it that worked really well and parts of it that were pretty messy at the time.
Yeah, definitely. Like we were really trying to figure out how best to work with different cell types and how to analyze the
Mm-mm.
data. I mean, I think the wet lab side, we'd do an experiment and we'd get 60 to 70 cells and be really pleased with that number of cells. I
Right.
mean, you look now and experiments are in the millions of cells quite easily, but back then, just the number of cells and how you do those experiments, or how you do them by sorting cells into individual wells of a plate with flow. And then on the other side, the analysis, we're really lucky now to have had an ecosystem of tools written by incredibly talented computationalists around the world to analyze single cell data. These are very complex, large data sets. And so,
Hmm.
but back then we were also figuring out, well, what is the best way to deal with these big matrices and
Right.
to extract biological information out of them, and kind of writing things from scratch. And,
Yes.
it wasn't the case you just go and download a package that thousands of people had used. You were sort of writing your own methods and
Hmm.
benchmarking them and figuring them out.
Yeah, which, of course is laborious, but I guess it allows you to really get under the skin of it as well and really understand it.
Yeah, absolutely. Like, I think everyone at that time really understood what was going on and
Mm.
was deeply involved in all of it, and that was just really fun to
Yes.
see, and to looking back, to be at the birth of this new technology. And then also, as with Sarah, she founded the Human Cell Atlas along with Aviv Regev, as the two co-founders of the HCA, and just this international consortium to understand every cell type in a human, and being there at the sort of the start of the technology being used and the start of this project that was going to use this technology to massively advance our understanding of human biology. It was really exciting.
Yeah, absolutely. And so you spent several years there and moved over to a short step to the Sangha. And then at some point, there came a decision to maybe go and apply some of these things, I would guess. But tell us a bit about that transition from academia into industry and how that came about.
Yeah, absolutely. So I'd been working on academic projects, working on the Human Cell Atlas. I'd always been interested in that sort of intersection of entrepreneurship and science and
Mm,
commercialization of science, and working in industry. So it's just a thing in the back of my mind, thinking about what could happen. I got offered a group leader position at an institute in Oxford.
hmm.
To go and kind of pursue academic interest in the T cell receptor sequence space, which was really very appealing. I turned it down mostly because I realized that I didn't actually want to go up the academic pyramid like
Right.
that.
Yeah.
We just our first daughter had just started school. My wife was very pregnant with our second child. My wife had a job in Cambridge that she really liked,
Hmm.
and although we could totally have moved to Oxford and made it work, it felt like a big upheaval for a career that, when I sat down and thought about it, wasn't really what I wanted to do. Like, it
Mm.
wasn't, I didn't feel that, like I said, when I didn't do my PhD after my master's, I didn't feel that burning desire to end up as a professor or a
Right.
senior PI. And I felt like the difficulties of that position, those jobs, which are, they're very hard jobs to do. And
Yeah.
I don't think, if you don't feel like that burning desire, it's probably not, that's probably a signal that you shouldn't pursue them, right? So
Yes.
decided not to do that. Actually, it was just after turning that job down that I started working with Sarah on the Human Cell Atlas. And then, yeah, after 18 months working on the HCA, realized, okay, it is time to move on from academia. It's time to join industry. And we'd been working with the 10x Genomics platform, the sort of first commercial droplet-based single cell platform that had
Hmm.
come to Sanger around that time. Sanger, of course, was really good at having the new toys, all the new
Right. Yeah.
instruments and sequencing instruments. So we'd been generating data from it. I thought 10x was a really interesting company, building interesting products, and also building products in immunology that were very well suited to what I'd been doing, kind of as a scientific interest. And so, yeah, I was really fortunate enough to be able to join them as a computational biologist, and also not move to California. I was just talking about, it wouldn't have worked for me to move my family to Oxford. It
Yeah.
definitely wouldn't have worked to move my family to the West Coast and the Bay Area, which is where 10x were located.
Fair enough.
But we agreed that I'd be able to stay in the UK and work remotely. And obviously, as a computationalist, that's much easier than having to run experiments in my kitchen, for example. So that worked really well. And yeah, and joined them and spent 4.5 years building single cell products. Working on developing immunology based products with 10x, doing a bunch of other things that I could do kind of remotely from them. yeah, ended up as a director in computational biology there, leading
Mm.
a team thinking about extracting biological information from single cell data, and also leading a product team building one of the products that we launched.
Yeah, interesting. And I'm guessing, given the stage the technology was at, and correct me if I'm wrong on this, but that although it was a commercial role in a commercial company, still a very research focused position and group and things like that. Was it, from what you remember, was it very different from academia? What were the similarities? What were the things you noticed that were different? Tell us a bit about the transition.
Yeah, for sure. So absolutely, it was very research focused. I
Mm.
mean, I joined that company when it was about 250 people, and I left when
Hmm.
it was 1,200-ish and had gone public and gone through the IPO. So I was,
Right.
had saw a lot of growth there. But yeah, very research focused, very much interested in doing good science, asking proper scientific questions, doing good experiments. So I think that sort of curiosity and that intellectual desire to find things out was the same. I think the incentive structures between industry and academia are very different. In academia, you need that next paper, either ideally as a first author or a last author. You need that next grant,
Yeah.
and you're working always towards that. And in industry, well, in a product company, certainly like in a tools company like
Hmm.
10x or like we are like in CS Genetics, you're working to get a product out that enables science and is really performant and is really good and works for customers and delights those customers so that they buy more of that product. So you bring more money in, so you can do more science to build new products. And
Yes.
that's a very clear incentive. And I think it really motivates teamwork across the entire organization, or to do it well, I think you have to
Hmm.
motivate teamwork across the entire organization. Academia can sometimes be a bit of a zero sum game. I get this grant, so you don't, or I
Right.
scoop you on this paper, so you don't get to publish.
Hmm.
Um, Whereas I think within industry, it's very, when it's done well, it's very collaborative, it's very collegiate, and you're all pulling for that really clear goal that's,
Yes.
measured in, did we sell this product? Was it
Yeah.
successful? Does it meet its specs, and are you selling it?
No, I see. And that journey that the company went on while you were there, obviously that's kind of what people hope for when they join a company, that it goes on that trajectory. But of course, in terms of the size, but also in terms of going public, that's quite a lot of change I expect happened within the business. What were your experiences of that?
Yeah, I mean, it was very exciting.
Mm.
Seeing, product launches were exciting. Gearing up for the IPO, I wasn't senior enough to be involved really in any of that, but hearing, we're going to we were going to have the IPO, we were doing the roadshow, and then, watching the live stream from the Nasdaq when they rang the bell and it went public, and that was, yeah, I think it is what people are hoping for, and
Mm.
I hadn't necessarily expected that to happen when I joined. I was just sort of interested in doing interesting science and,
Yes.
having a job that was fun. But yeah, seeing that was a really nice sort of ancillary benefit, and yeah, really motivating and exciting.
Mm. No, I bet. And I bet that the stock exchange part of it was quite the party.
Yeah, it looked like, I think the people who went to New York for that had a fun time.
Yeah.
I had a little glass of champagne in my home office at the end of our garden.
Fair. I've just got this image of the live stream stopping and someone going, right, back to work. So tell us then about the transition from that through to your next role and eventually into CS, and how you ended up there.
Yeah, for sure. So I said that I got to stay in Cambridge,
Mm.
which was fantastic for my, family life, work-life balance. Like I said, I couldn't really have moved to the West Coast for various family reasons. So that was great. But the time difference was always pretty hard.
Right.
8 hours behind, or if, being 8 hours in front of all your work colleagues makes scheduling meetings pretty challenging.
Yes.
You have to do evening meetings. And as I got kind of more senior and had more and more meetings, and meetings with larger teams or meetings with,
Mm.
with leadership, there wasn't really any way around having evening meetings. I'd have people on my team who'd be happy to meet at 7 a.m. their time, for which I was always really grateful, but there were always meetings at, 8 o'clock UK time, 9 o'clock UK time. And I would, at the start of the week, I'd email my wife with my schedule for the evenings and
Right.
say, I'm going to be in meetings on these times on these days. I might have dinner with the kids on Wednesday.
Yeah.
or do
Yeah.
bedtime on Thursday or whatever. And I don't regret any of that at all. But it got to a point where that was getting wearing me down a bit. And
Mm,
I think also the pandemic accelerated this because I had
okay.
2 years of not going out to the West Coast to see people. So I working at home is great, but I really, really valued those weeks every quarter or so where I'd go out and go to HQ and chat
Yes.
to people and rebuild those connections and see people. And 2 years of not doing that also I think probably
Mm,
accelerated my desire to not be so far away in terms of time difference and actual miles. So I started
yeah.
thinking about what was going to be next. When 10x had the IPO, they had photos of the sort of history of the company. And there were photos of the original team in a literal garage in the Bay Area. And
Yeah.
I thought actually it looks pretty fun to try and build something from essentially scratch or nearly scratch and to be involved in those early days of building
Hmm.
something cool. So I looked around, spoke to my network, and there was a company, a small startup in Cambridge, who were pre-seed, and they were an AI-focused company looking at T cell receptor sequences. So I joined them as head of biology, worked on kind of the plan for generating experimental data. They were sort of 9 people when I
Mm,
joined. We worked on raising our seed round, which was challenging, but like a super good learning experience for me on how to work with
yeah, I bet.
investors and
Yeah.
pitch investors and think about raising money. And I spent 9 months with them. It wasn't exactly the right fit for me, I think, in terms of what I wanted to do.
Mm,
And I sort of learned that CS Genetics were looking for someone to come and lead R&D. And actually, I've known the founder of CS Genetics, Luke, for since we did our PhDs in the same institute. So Luke
oh, I
actually
see. Okay.
sat next to me in the office in the Babraham Institute. He was in a different lab, but we shared an office. So I've
Hmm.
known him for a long time. I saw him found the company. Always kind of interested. I'd go for coffee with him, and obviously, when I was at 10x and he was working on CS Genetics, we never talked about work because
No.
there were conflicts and or potential conflicts that we both really wanted to avoid. But we would just say, how's it going? And I was speaking to him, and he said he was looking for someone to come and lead R&D at CS Genetics. And
Yeah.
I thought actually I really, really enjoyed building tools. I really enjoyed building tools as an academic when I wrote software that went out into the world and other people used to answer their questions. I really enjoyed building tools at 10x where we built products that people used. And it's a real effort multiplier. You put the
Mm,
work in, you send it out into the world, and then tens, hundreds, thousands of people use that work, the
yes.
product of that work, to answer questions across all of biology. And that really resonates with me. I really, really like that as a thing to do. And sort of I thought, well, CS Genetics felt like this amazing example of building a tool and building a tool that had incredible potential and was really interesting
Mm,
in a space that I understood, both as a customer and as someone who had thought about what products look like in this space. Um, in a team that looked, he told me about the team, and they are just phenomenal, incredibly smart, talented, nice people.
right.
Yeah, it just felt like a really good opportunity to come and be very early in a company that was going to build really very impactful and exciting tools for other people to use to answer questions in biology.
Yes. And what stage was the company at when you joined then?
So they'd raised their Series A
Yeah.
a few months before I joined,
Okay.
and were sort of working on. So that was almost exactly a year ago, and we're working on getting ready to launch the product.
Yeah, I see. Okay. And you've talking through your career, you've been in large academic institutes, into commercial company that started reasonably small, but not tiny, but then grew pretty significantly, into very small companies post that, and in roles of increasing seniority as well. From a career perspective, I suppose, or in terms of perhaps the key skills that you've learned along the way, or the things that you've had to develop along the way, what have been the most important learns, do you think, for you?
Yeah, good question. I mean, I think one of them is accept that luck's going to play a really important part
Mm,
in what you do. When I started my PhD in this area of B cell receptor sequencing, it was a little bit of a sort of niche area of immunology that was fascinating
right.
and weird. But during the time I was doing it, cancer immunotherapy became more and more important. So actually caring about how the immune system sees antigens became incredibly translationally important.
Mm,
And joining Sarah's group at the start of the single cell revolution, there's obviously some luck involved in these
yeah.
steps, and being able to kind of work on the human cell atlas. So luck's super important, but you can also augment that luck by giving yourself maximum surface area to receive luck, I suppose. So be in those,
Right.
be in those meetings, go and make sure you talk to people at every opportunity, take the opportunity to meet with people, to go
Yes.
and put yourself out there. So I think, harness luck and drive it as best as possible. I think another advice is like, know you will know internally, and I think subconsciously, when it's time to move on to something else for
Right.
whatever reason. There will always be times in a career where you start to think, now is the time that something else is would be better for me, for whatever reason. And I think
Mm,
listen to that, and don't don't hold on to something for too long. If you think it's the right time to move on, do that in the most professional way possible with a nice
yeah.
transition. Make sure you're doing it right, but also listen to that, and do the thing that's right for you, and your what you want to do next. So I think that's
Yes.
really important. And also be kind. I think it's super important. There's that quote that goes around Twitter, everyone here is smart, distinguish yourself by being kind.
Right.
And Sarah had a lab motto, which was be bold, be brilliant, be kind. And I think that's possibly underappreciated in
Okay.
science generally. I think I've worked with very kind, thoughtful people across academia and industry. And I think, yeah, just it's easy to think of science as a zero sum game. It's easier to think of
Mm,
it in a way where someone has to lose to win. But I don't. I think it's possible to have a good career where you do really good science and make cool things, whether those are papers or products or drugs,
yeah.
but still be kind to people. Make the kind decision if you have two that are kind of otherwise equally weighted. So I think that's really important.
Yeah, absolutely. And so, and I think particularly in academia, it's challenging, right? Because there's so much emphasis on you getting your name as far up the author list as possible and on the lab and all that kind of thing. And it's easy to see that, as you say, as a zero-sum game and at the expense of someone else. What do you think in those circumstances then? What does being kind actually look like?
Good question. I think it's making sure, especially if you're in charge of a team,
Mm,
it's more impactful if you have other people, if you're responsible for other people. If
yeah.
you're an individual contributor in a more junior position, being kind means, helping people be a decent member of the lab, do your share, make sure you share knowledge and resources as appropriate. I think when you're more senior and looking after people, it's making sure that your team have what they need to succeed on the terms that they want to succeed on. Make sure they have the resources to develop in their career. Make sure you support them. Obviously, you want to support them mostly in what they're doing for the company, but they're also people and they have needs and what they want to do. So making
Mm,
sure that they feel supported in a more holistic way. I think both Anne, my PhD supervisor, and Sarah, my postdoc supervisor, were incredibly good at supporting my career and helping
right.
me talk through decisions, helping me think about what was next, giving me advice. Sarah was, when I told her I was going to go and interview at 10x, she said, "That's an amazing opportunity, fantastic, well done," and was just so incredibly supportive. You hear these
Mm,
stories of academic supervisors who, when you say you're going to leave academia and move to industry, they don't talk to you again or tell you that's a
yeah.
terrible idea. Why would you do that? And I've never experienced that. So
Yeah.
I think, just trying to make sure you, like, even outside of science, make sure you think about what the other person needs and how best to help them with that.
Yeah. Do you know what? It's something that we see a lot when people move out of academia, and it is a shame that, there's not everybody by any stretch, but there are a group of people that anybody leaving the lab, you can't do this. How could you do this to me after everything I've done for you? These sorts of things. And it's a real shame that happens. I think, it always does make me laugh as well that they talk about these alternative careers when 75-80% of people end up doing them. But there you
Yeah,
go.
for sure.
That's a different conversation. Um, I really like this idea of giving yourself the greatest surface area for luck. Um, and you talked about sort of being proactive with that and putting yourself out there a little bit. Can you talk a bit more about that and how that's worked for you?
Yeah, sure. So I think I guess it's hard to unpick how much of this is luck, but it's always saying
Mm,
yes to the meeting, right? Like I've never regretted giving a talk.
okay.
I've always found an invitation to go and speak or an opportunity to speak, whether that's, in your grad school symposium and you can go and give a
Mm,
talk. Put yourself out there. It is incredibly good practice every time you do it. Every time you give a talk, if it's not important, it's just for the actual talk. It's just practice at doing it. And that's
right. Yeah.
such a good skill and will put you, as a junior scientist, will set you in such good stead if you can present your work well and engagingly and make sure that people follow it and like it and come away with a good
Mm,
impression. Or at a conference, or if you're invited to come and speak, always try and say yes if you can, because that just,
yeah.
that does increase the number of people who've seen you and recognize your name and might think of you when they're doing something. I guess say yes to meetings. I when I was at Sanger, I was invited to join a meeting with some of the 10x leadership when they came in the early days to come and talk about products because it
Mm,
was relevant to work I was doing. And that, started to build connections with people at the company, which were useful later when I was looking to go and join there. So I think
right.
just always trying to interact with as many people as possible, get your work out there, make sure you practice getting your work out there, and people know what you're doing and then can think about you for next time they're looking for someone or a collaborator as an academic or whatever.
Yeah, I think it's such an important point because I think, especially when you're doing your postdoc or your PhD or something so intense like that, the science is all consuming, right? And it can be tempting to see anything outside of that as non-core or
Mm.
distracting or what have you. But your project is going to finish one way or the other at some point, right? And you've got to think about what happens next. And some of these things you don't get a direct return necessarily, but you
Mm.
never quite know when it's going to come back around.
Absolutely, and it's always possible to think, oh, I don't like doing public speaking. Maybe I'll just
Mm,
choose to do the poster, or I'll decline this because it's difficult. But it really does. It's a virtuous cycle. Every talk you give, the next one's easier, both because you've practiced it, and so you're just kind of more comfortable, but also because you've made all the material. If
right.
you're doing a project, you make the slides once, and then you can use them in multiple situations. So it's really worth doing.
Yes. Yes. And then sometimes some people one day get to the point where they can turn up and 5 minutes before go, which talk is this? Oh yeah, okay. This.
Absolutely.
Yeah. And not all of us get there, unfortunately. Um, so going back to CS Genetics, you touched on this a little bit a few minutes ago, but tell us a little bit about what's next. What does the near future hold?
Yeah, absolutely. So we're right now just finalizing the final commercial version of the product that we're going to
Mm,
launch. So it's going to be a single cell gene expression assay. Um, we're working with some very early access customers
right.
locally and further afield, and we're getting some really quite exciting data back from them. I was looking at some earlier where there's some pretty cool biology that we're revealing from our approach, which is great, and I find that very motivating just to
Mm,
see, look at the data and see biology, because coming back to this idea of building tools and revealing biology, it's just great. So we're working on that right now. We're gearing up for launch later in the summer. Going to get that first product out there and shipping to people. And then working on our roadmap and
yeah.
building out our product suite to address different needs. Very excited about the idea of scale. I said, the product scales from single sample up to thousands of samples. The first kits are going to be in 8 and 16 sample format,
Okay.
but I'm really, really excited about delivering this idea of many, many samples in a single cell experiment. Right now, a lot of single cell experiments are a lot of cells,
Right.
but not necessarily a lot of samples. But I
Yes.
think there are questions you can ask with a lot of samples that are really fascinating and people aren't really doing right now. So enabling that, and we're already talking to some potential customers who are thinking about thousands or tens of thousands of samples, maybe even hundreds of thousands of samples. And so
Okay.
thinking about how we deliver that in a way that's useful for customers and kind of unlocks this whole new world of single cell. That's really exciting. So going to work on that. hopefully continue to grow, continue to, yeah, just build cool stuff,
Yeah, yeah.
and just keep with that idea of we're building tools to help people do science.
Yes. And that's the key thing, isn't it? Is, it's a really exciting point, but this idea of sort of democratizing single cell and giving more people access to single
Mm.
cell. What are you hoping will be the ultimate impact of that?
Yeah, I mean, I'd love to see a lot of people using our product to do things that they're doing in other ways right now, where
Right.
they're doing experiments where they are they're measuring something that's obviously useful, but they can just do essentially the same amount of work for not a great deal more cost, but
Mm,
suddenly reveal a huge amount more information about their system, and hopefully that accelerates drug discovery. I would love to be in a world where a drug comes to market that was in part powered by our product and maybe accelerated by our product. That would
yes.
be, that would feel extremely validating and exciting. And also just seeing more and more papers published, scientists using it to unlock possibilities that they couldn't do either because of
Yes.
scale or cost or workflow. And yeah, I think that's going to be where I'd like us to get to.
Fantastic. Well, we wish you the best of luck, of course. We'll be watching with great interest, and yeah, thank you so much for joining us, Mike.
Well, thank you, Tom. It's been really fun.